crossroads of four biomes. The deciduous forest to the east, the needle-leaved evergreen forest of the boreal zone to the north, the prairies of the Great Plains to the west, and the oak savannas of the Midwest itself. Its glaciated landforms harbor many wetland communities, and the wave and windswept shores of the Great Lakes support beach and dune communities like those on the Atlantic coast. Human activities have disturbed, fragmented, or destroyed natural vegetation on a wide scale, with over 99.9% of the original prairies and savannas now lost. Wisconsin thus provides outstanding opportunities for studying trait and trait-environment correlations, environmental determinants of species distributions and phylogenetic community structure, and drivers of local species extinctions and explosions, given its diverse flora distributed across a wide range of climatic and soil conditions, large numbers of introduced plants, and unparalleled data on shifts in species distributions and abundances over hundreds of sites over the past half-century. So Don Waller and I and Ken Cameron and Ken Sytsma, our students and postdocs, are studying these issues, focusing on drivers and consequences of functional, genetic, ecological, and phylogenetic diversity in the Wisconsin vascular flora, with financial support from the NSF Dimensions of Biodiversity program. These studies hinge on developing a molecular phylogeny for the state's flora, scoring several functional traits across the flora and selected species representing forest and prairie communities, modeling determinants of species distributions, evaluating trait and trait-environment associations, analyzing drivers of species turnover at hundreds of sites, and quantifying the current and expected future impact of climate change on species distributions. Today I'd like to illustrate some of our research by using our Wisconsin phylogeny to test several hypotheses about how floral, fruit, breeding system, and life history traits should differ between native and introduced species, and how they should vary with each other and with habitat openness in native species. Postdoc Ricardo Kriebel did a masterful job conducting tests and management of the data. He and I tabulated the trait data, and I proposed the hypotheses to be tested. Former postdoc Daniel Spalink, former grad students Chloe Drummond, Jeff Rose, and John Zaborsky helped assemble the phylogeny in the Sytsma and Cameron labs. We estimate that the total angiosperm flora of Wisconsin is 1,595 native species and 851 introduced species, representing roughly half of the flowering plants of the northeastern US and adjacent Canada. Native species are distributed in largely log linear fashion among 122 families and 517 genera, with 7 families and 9 genera having unexpectedly large numbers of species and accounting for 48 and 21% of the total native flora, respectively. We have been able to include in our molecular phylogeny 1,568, or 98% of the native species, and 790, or 93% of the introduced species. This phylogeny is based on a RAXML analysis of RBCL, MAC-K, and ITS sequences obtained almost exclusively from vouchers at the Wisconsin State Herbarium. We constrained 41 nodes in the phylogeny to accord with relationships inferred from studies involving a greater sampling of sequences or taxa. Now, for this project, we assembled data for all species in the phylogeny for origin, native or introduced, growth form, herb, graminoid, shrub, tree, or vine, life history, annual, biennial, perennial, or various combinations thereof, flower color, white, cream, blue, red, green, or brown, petal or follicular length, pollinator, biotic or abiotic, breeding system, hermaphroditism, monoecious, dioecious, or other, mode of seed dispersal, fleshy fruits versus others, occurrence in open, intermediate, or closed habitats based on presence or absence, and flowering season during either the sunny or shady phase of understory communities. This trait set includes over 28,000 entries. Species in each category within a trait are mutually exclusive, except for occurrence in open, mixed, and closed habitats where smaller sets of species occur in 2 or 3 of these habitat types. Open habitats have the largest number of species and closed habitats the least. Now, our hypotheses regarding trait environment and trait associations are based either on the classic literature or some of our own ideas. We predict that introduced species, by being associated with human disturbance and transport, would have a greater frequency than natives in annual life history, herbaceous growth form, occurrence in open habitats, visually conspicuous flower colors, which should be associated with life in open habitats, strongly illuminated by broad spectrum light, larger flower size, hermaphroditic breeding systems, which would be associated with larger, visually more conspicuous, and therefore more costly flowers, and passively dispersed seeds due to a greater association of fleshy fruits with shaded habitats that are largely windless. Introduced species should also have a lower incidence than native species in mixed and closed habitats. Most of these hypotheses are strongly supported by the data. Tests were based on analyses of phylogenetic correlation and contingent evolution implemented in phytools, as well as phylogenetically structured t-tests and ANOVAs tested using custom R scripts. Introduced species, as predicted, have a significantly higher fraction of annuals. You should see 40.1% versus 14.4% of native species. Of herbs, 72.9% versus 59.5% of native species. Of more conspicuous flower colors, meaning white, blue, red, and yellow, versus cream, green, brown, or absent as inconspicuous. Higher incidence in introduced species of larger flowers, of hermaphroditic flowers, and of occurrence in open habitats. Introduced species also have a significantly lower incidence in closed habitats, 4.1% versus 24%, and mixed habitats, 25.6% versus 46.0%. Interestingly, among graminoids, grasses have a much higher incidence of introduced species than other clades, and we suspect that this is mostly due to deliberate introduction of many grass species for forage, and surprisingly few species of sedges or other graminoid groups. Tests for contingent evolution have also revealed several patterns that concur with our hypotheses. For example, we found that life history and origin were dependent on each other in the best model, and the reconstructed transition rates were consistent with species accumulating in the annual introduced and the perennial native vegetation categories, which are circled here. We use the same approach to test hypotheses regarding trait and trait environment correlations with the native flora alone. For trait correlations, we predicted that hermaphroditism should be more common in animal pollinated species, that unisexual flowers should be smaller and have less conspicuous flowers, and that dioecious species should be more likely to be woody and have fleshy fruits, bear small, inconspicuously colored flowers, or have abiotic pollination. Our data support these ideas. Hermaphroditism is more common than unisexuality in animal pollinated species. Flower size is smaller, and conspicuous colors less frequent in unisexual versus hermaphroditic flowers. And dioecious species are more frequently woody than hermaphrodites. They more frequently bear fleshy fruits, and they have flowers that are smaller and more frequently are inconspicuous in color. All these patterns accord with our prediction, and as you can see, almost all of them are statistically very highly significant and explain substantial variation in trait distribution. Now, based on my 1982 model involving how the higher advertising costs of attracting and dispersing pollen in unisexual versus hermaphroditic flowers should increase with flower size, and how seed dispersal might increase with total crop size, in species with fleshy fruits versus passively dispersed seeds. Dioecious should be common only in plants with small flowers and fleshy fruits. Monoecious should be common only in plants with small, often wind-pollinated flowers and passive seed dispersal, and hermaphroditism should be common in plants with larger flowers, regardless of seed dispersal mechanism. Our data provide the first support for these patterns based on quantitative variation in flower size. First, the incidence of unisexual flowers, as you see, drops with petal or ligule length with a log scale on the x-axis there, and that of hermaphrodism, of course, rises, as shown by these highly significant logistic regressions. The pattern is stronger if grasses with their minute and wind-pollinated, but often hermaphroditic flowers are excluded. Second, the incidence of dioecious increases with decreasing flower size, to the right here, to the left, pardon me, in species with fleshy fruits, and is essentially absent as predicted among those with passively dispersed seeds. Third, as you can see, the incidence of monoecious increases with decreasing flower size in species with passive seed dispersal and is essentially absent in species with fleshy fruits. So taken together, the results of this and the previous slide strongly support my 1982 predictions regarding the importance of ecological drivers of plant breeding systems. For trait environment correlations, we predicted that in open habitats, flowers should be larger and have more conspicuous flowers than elsewhere, that fleshy fruits should be less widespread, and that dioecious should be less common. In closed habitats, the converse proposition should be true. The data support 5 of these 8 predictions, 4 in highly significant fashion. The 3 exceptions, highlighted in red, are that there is little difference in flower size between open and non-open habitats. There is significantly higher, not lower, incidence of dioecious in open habitats, and conversely, there's significantly lower but not higher incidence of dioecious in closed habitats. The first exception reflects variation among species in biotic versus abiotic pollination and the width of their distribution along light gradient, time of flowering, and growth form. It disappears when we focus on animal pollinated herbs occurring in open habitats only, right, versus those occurring in closed habitats only, and separate the latter into species flowering in sunny conditions under open canopies before the canopy closes versus shady conditions under closed canopies in midsummer. As I predicted in 2002, herbs of open habitats like many gentians or which flower under open canopies like many trilliums or violets have large, visually attractive flowers, while those which flower under closed canopies like tway blades and wintergreens have smaller, less attractive flowers. Among all species with animal pollination, flowers are significantly larger if they are blue, red, pink, orange, or yellow than if they're cream or white, and all are significantly larger than green or brown flowers. Again, it's expected from theory and the distribution of larger flowers in more brightly lit site. The failure of the 2 dioecious environment hypotheses can be traced to my having ignored the fact that wind pollination is and should be more common in more open habitats, where it accounts for 46% of the species versus 23% in closed habitats. The strong tendencies toward dioecious in wind-pollinated species and the tendency toward wind pollination in open habitats are great enough to override the expected and observed association of dioecious to small flowers and fleshy fruits in the animal pollinated species. And so even this exception, the original exception to the expectations is illuminating, and you can see that the Wisconsin angiosperm flora supports most of our hypotheses regarding trait differences between native and introduced species, trait-trait relationships, and trait-environment relationships. My colleagues and I would like to thank the NSF Dimensions of Biodiversity program for their support of this and related research, and I would like to call your attention to a new postdoc position available in my lab to study phylogeny, historical biogeography, and floral eco-evo-devo in California. If you're interested, beam me an email. Thank you.